First of all, I would like to thank the organizer for inviting me here. I am Rajat from Dr. Dimple Notani's lab at NCBS, and I'm going to talk about how RNA provides specificity to condensate and transcription factor binding. So my work would be majorly focused around these proteins called transcription factors, so that recognize and bind to certain sequences in the DNA. So what it does if the sequence is exposed, by meaning exposed if it is not embedded inside the nucleosome, then transcription factors are proteins that are known to bind to these sequences, and they recruit different 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 activators like P300 mediators, and as well as the polymerase machinery to the site, which subsequently lead to the transcription of the said gene. Now, what I wanted to understand is how does the binding of the what are the different factors that regulate the binding of transcription factors? So, in order for the cell to regulate the transcription inside the cell, it is important for the cell to regulate the binding of these transcription factor. So, first is the motif itself. So, transcription factor doesn't bind to a single motif; it binds to a consensus of different motifs at variable strength. So, the strength of the motif is how similar that particular region is to the consensus motif. The higher the similarity, the higher is the strength, and the higher is the binding of the transcription factor. So, in addition to this sequence, the DNA information contained inside the DNA, there are other factors which can also regulate the binding of transcription factor, which would be the focus of my talk. So, two of these factors are first the transcription. How does the process of transcription in turn regulate the binding of the transcription factor? And the other is how does the output, that is the RNA itself, how does that regulate in controlling the binding? So the model system that we have used is an estrogen signaling system. Estrogen is a steroid hormone which can translocate from the extracellular milieu to the inside of the cell, where in the cytoplasm it recognizes and binds to the receptor called ER alpha, which is the transcription factor in question that we are going to talk about. And upon binding to the receptor dimerizes and it translocates inside the nucleus, where it binds to its own motif called the ERE, or the estrogen response element, and which, like other transcription factors, it would lead to the transcription of these genes. Now, what I wanted to understand is how transcription as a process feedbacks into this. So transcription is a multi-step process that requires initiation, then followed by elongation, and then leading to termination. So what I wanted to understand is, if you perturb transcription, how does the binding of the transcription factor gets perturbed? So we use this particular drug called DRB, which is a elongation inhibitor. So what DRB does is it would prevent the transition from the initiation to the elongation phase. So the initiation rates are the same, and that would lead to the increased accumulation of polymerase to these sites. So which, after treating the cells with these DRB, there would be an increased amount of polymerase to these sites. Now that along with E2, so first I treat the cells with DRB, which is the inhibitor, then to stimulate the binding of the transcription factor, after treating the cells with DRB, I add E2 to the mix. And after 1 hour of E2, so every E2 has E2 signaling system has a peak, and that peaks come as around 1 hour. So at 1 hour, majority of the transcription factor is bound to the DNA. Now, what I wanted to see, if you perturb the transcription using this drug by increasing the polymerase at these sites, how does the binding of the transcription factor gets perturbed? So to look into to assay the binding of transcription factor, we employed this technique called ChIP-seq. So ChIP-seq tells you where exactly in the genome your protein of interest is binding. And what you see here, the end product that you see in these regions are the peaks, or the regions that are the binding sites that are for ER alpha. These these sort of triangle-like regions are the binding regions for ER alpha, and this is on one of the responsive genes called GREB1. And what we see here is the binding of ER alpha after treating the cells with DRB is increasing. Now we wanted to see genome-wide, what is the effect of the binding genome-wide. So we looked at all the binding events. We see that there is a gain of around 2000 peaks and a loss of around 1500 peaks genome-wide of ER alpha. Now we wanted to see how does the binding profile of these peaks look like. So now we looked at these 6500 peaks and these 2000 peaks. So what you see here is a summary plot by summarizing the counts from all of these common sites, the 6000 sites, and what we see here is that the binding of ER alpha on these regions are going up, which is on the common sites, and as well as there is an increased number of gain sites as well. So combining both of these, the increased binding on the common site as well as an increased number of gain sites, suggests that there is an increased binding of the transcription factor genome-wide. Now what we wanted to understand is what regulates the binding of the transcription factor now, after treating the cells with these inhibitors. So we already knew that motif is one of the things. The information contained within the DNA is one of the feature. So first we looked at the regions. So these are all ER alpha peaks, and all the ER alpha peaks are binned on the basis of the motive strength. So high motive strength would mean that it is more similar to this ER alpha ERE motive, and low meaning it is less similar. And within each bin, the motive, the underlying information is the same, but one thing that is varying is the polymerase levels. So 1, 2, 3, 4 means the increasing level of the polymerase. Now we wanted to see if we normalize one variable, that is the DNA information contained within the DNA, how does the binding of the transcription factor after you treat the cells with DRB, how does that relate to the polymerase level now? What we see in the same bins that, so this is log to fold change values for DRB over DMS row, quantifying the increase. And what we see here, the regions that showed the highest pol2 were the regions that were showing the most gain. So wherever there is a higher increase in the polymerase, those are the regions that are showing a higher increase in the ER alpha binding. So to summarize, what we have seen here is, in a normal situation, transcription factor would recruit the polymerase, and that would lead to the transcription of the cell. Now, after you treat the cells with these inhibitors, what would happen is there would be an increase in the post polymerase because of the drug, and now which would lead to increase in the transcription factor binding. And where exactly transcription factors are binding, these are the regions where already had a higher polymerase level. Now, the ChIP-seq experiment tells you a population, like if you take millions of cells, what is the output from the millions of cells. Now, what we wanted to see, what actually happens at a single molecule level. So we use this technique called single molecule tracking experiments. So what you do in this, you take the protein of your interest, you label it with the given tag like halo tag and that, so that you can vary the dye concentration, so as to get a very less number of particles labeled. And from that very less number of particle label, you can get the trajectories of those particles. And from those trajectories, you can get a rough idea of how fast they are moving, how if they are diffusive, if they are moving within a confined population, or other factors. And other thing what you can also calculate is the residence time. The residence time tells you how long a given transcription factor stays at a given region. So we did the same thing with ER alpha. This is ER alpha labeled with halo. We did it along with Gunjan from IIT Hyderabad. And these small blips that you see are the single particles of ER alpha. And we did these two experiments in both DRB and DMS to quantify how does the motion of these particles change after you treat the cells. So what we find here, so unbound fraction is the diffusive fraction, the one that is moving freely, and the bound fraction is the one that is subdiffusive. It's not moving freely, but it is something is perturbing the binding of the transcription factor from moving it freely. So what we find is that in the case of the unbound fraction, we see that there is a decrease in the unbound fraction, and there is simultaneous increase in the bound fraction. This is similar to what we see with ChIP-seq as well, the population level, that at the single particle level, we see that the bound fraction of ER alpha that is bound to the DNA is increasing. Now we just wanted to see what exactly is happening with the bound fraction, the protein that is actually bound to the DNA. So we perform the same imaging experiments at 200 milliseconds, because at 15 milliseconds, what you do is you keep the laser power so high to get the particles, but what can happen is these particles can get bleached. So to get a better idea of the transcription factor motion, so we did it at a lower frame rate of 200 milliseconds. And what we did is we calculated the dwell time. And from the dwell time, we did it in both the DMSO as well as in DRB condition. And from the dwell time, we get a survival plot of how long a given particle is staying at a given time. And we see that majority of the particles are staying at a given location for a lesser time, but as the time increases, there are few particles. And we fit this into two different population, a bi-exponential fit, and what we get is two different population. First is the population that shows the dwell time of around 2 seconds, and the other is the population that shows the dwell time of around 10 seconds. The population that shows the dwell time of 2 seconds is the transiently bound fraction, which is bound to the DNA for a lesser time, and what is bound to the DNA for a longer time, that is of 10 seconds, we call that as the stably bound fraction. So after treating the cells with DRB, what we find is that the dwell time of the transcription factor went from 10 in DMSO to 13, that is in DRB, and for the transiently bound fraction as well, it went from 1.9 to 2.906. It's a very subtle change that we see. To summarize, what we have found here is that as you inhibit the elongation, using DRB, what we see is the polymerase gets stabilized. And because of the stabilizing, we see an increase in the chromatin bound fraction. And once we calculate the dwell time for the CR alpha, we also see that there is an increase of the dwell time, which is sort of similar to what we see for what we saw for ChIP-seq experiments as well. Now, ER alpha is known to form these condensates. People have reported, us and others in the field have reported that so this is ER alpha inside the MCF7 nucleus. And as soon as you add the ligand, we see that there is a formation of these condensates here. And now what we want, so now all of a sudden why I'm talking about these condensates is because people have talked about that there are certain these condensates, especially for MED1, that is selectively is known to partition these positive regulators of transcription like RNA polymerase and other things, and which are in turn, people have shown that it is sort of related, showed a positive correlation with transcription. And another factor is that we us have previously shown that these condensates have a genomic basis. So what we had done in this study is that we have deleted one of the regions, and now we checked, does the condensate formation on those regions change? And we saw that after deleting the region that is known to bind to ER alpha, the formation on the condensate on those regions change. So we think that these condensates sort of have a genomic background. So wherever there was a higher binding, those regions, multiple of those regions are coming together to form these condensates here. And from the previous experiments, we saw that genome-wide, the binding of the transcription factors was increasing, as well as how long a given transcription factor stays at a given location, that was also increasing. Now we wanted to see how does the condensate dynamics change in these. So we labeled ER alpha with EGFP, and we followed it with time. So the time that you see is time post addition of the hormone. Initially, it's a homogeneous, the pattern is homogeneous, and after you add the ligand, we see the formation of these spots. We quantified the formation of these spots by two different ways. First is we calculated the number density. How does the number of these things increase with time? So we see that initially, since the signal was homogeneous, we didn't see many number of these condensates, but as soon as the time increases, we see that it sort of reaches a plateau phase. So the other thing that we calculated is the size of these condensates. So this is the average diameter of these condensates, and we see that that also sort of follows an exponential increase, followed by a plateau phase here. We did these two experiments after treating the cells with DRB, and what we calculated is first the number density. We see that we calculated two different parameters. First is the maxima of the curve, that is what is the maxima of both the curves here. And what we see is the maxima in terms of the number density is not changing much, but we wanted to see how fast are these condensate forming. So we calculated what is called as the t-half of these curves. So how we calculate t-half is by fitting an exponential curve in these, and from that we get an idea of the rate at which these things are increasing. And t-half is the measure of how long it takes for it to reach half of the maximal response, and we did it in both these conditions. And what we see here in the case of DRB, we see that the formation of these condensates are faster, as it is sort of evident here as well. Next, we looked at how the size of these condensates are increasing. We see that in the case of DRB, we see that the formation of these condensates are bigger. If you look at the max here, the max curve between the two, the DRB and DMSO, is quite different, and which is the same thing quantified here, that the max diameter between the DRB condensates, the ER alpha condensates in DRB and DMSO is different. But once we looked at the diameter t-half, that how fast the diameter is increasing, that didn't show us a drastic differences. So now what exactly is so with the confocal live cell imaging, what we get a rough idea of two different populations. First population is one that is freely floating inside the nucleoplasm, and the other that is actually bound to the DNA. So what these increase with respect to the t-half and the increase with respect to the size can be attributed to both the population. If it is because of the freely floating protein, or it is because of the ones that is actually bound. Now, since we were interested in only the protein that is actually bound to the DNA, so we treated these cells with this buffer called CSK, which is known to remove the freely floating population from it. So, and we performed immunostainings for ER alpha after treating the cells with this buffer, and just to get just the DNA bound fraction, we calculated these ER alpha condensates volume fraction, and what we find here, that in the case once we look at the volume fraction in the case of DRB, which is also showing a similar increase, which was showing in the case of the live cell imagings. So to summarize what we have found here is that in the case of DRB, since the dwell time which we saw from the single particle tracking experiments was increasing, which can lead to an increase in accumulation of the transcription factor, as well as other factors as well. The polymerase might also be present in these regions, and which lead to a subsequent increase in the formation of these hubs. So now what we wanted to understand is how polymerase is actually doing it. What is the mechanism? How does the polymerase lead?
Are you saying that a single condensate is just one single complex?
No, there would be multiple. It's just
Okay,
just a representation. I'm not saying that it's a single complex. There can be multiple of DNAs coming together. So yeah, coming back to what I was talking. So what I wanted to understand is how polymerase is doing these things. So there are two different mechanisms how polymerase can assist the binding of transcription factor. First is, like I told you before, that the transcription factor cannot bind to the motif if it is embedded within the nucleosome. So for it to bind to the motif, it needs to be exposed. So for that, something has to evict out the nucleosome to free the binding site for the transcription factor. And polymerase is one of those factors which is known to recruit remodelers and other chromatin remodelers to these regions, which can lead to eviction of the nucleosome and which can expose the binding site for the transcription factor, leading to the stabilization that we see in the case of transcription factor. The other thing that can happen is the RNA. The RNA, so many of the transcription factor, beside having a very well characterized DNA binding domain, also have a less well characterized RNA binding regions. One of the less well characterized regions is already talked about before, the RRGG motif here. So the transcription factors like having those RNA binding regions can in turn be present in the proximity of these the motifs, and which can lead to the feedback to these motif regions, which can stabilize the binding of the transcription factor. Now, it is already known that once you treat the cells with these inhibitor, that is the flavopiridol is another elongation inhibitor, people have shown that as the polymerase increases, so does the binding of the nucleosome remodelers also. And that in turn can also lead to the opening up of the chromatin, like I was talking about. And now this can lead to exposing the sites of the transcription factors or the motif, which can lead to the stabilization. But for the case of the transcription factor, so ER alpha, like other transcription factors, also have a less well characterized RNA binding domain. And this is an RNA immunoprecipitation study which we have done here that is known to interact with TFF1 eRNA, which is another eRNA present in the nucleus. And this interaction sort of increases once you treat the cells with E2. So ER alpha is another transcription factor which can bind to the RNA, but what the question still remains that now I am treating the cells with the inhibitor, so there shouldn't be any RNA because the elongation step is getting affected. So there shouldn't be any RNA coming out of it. So, but that is not entirely true. So there is a population called the short RNA that comes out. So this is the RNA that is shown here. So after you treat the cells with these inhibitors, what people have shown that there is an increase of this short RNA that comes because of the polymerase getting stalled. The polymerase secretes this short RNA because of not proper elongation. So this short RNA can in turn affect the binding of the transcription factor. So now what we wanted to see is we wanted to decouple the effect of the two. So we took a stepwise approach. First, we treated the cells with this another inhibitor called triptolide. Triptolide would degrade the polymerase and which would also lead to a decrease in the levels of the RNA as well. So both RNA is not there and the transcription factor is not there. Now we assayed the binding of the transcription factor using ChIP-seq, and what we find here is the binding of the transcription factor goes low. So this is the same thing. So these are ER alpha peaks and the counts is shown here. So the higher the blue means the more the loss in these, the higher red would mean there is a gain. And we see that after treating the cells with TRP, that is the triptolide inhibitor which perturbs this, we see that the binding of the transcription factor goes low. So in the condition when there is no RNA, no polymerase to open the thing, we see that there is a decrease in the binding. Now what we wanted to see, what would happen if you were to increase the level of RNA without affecting transcription. So how you do that is there is a protein that is known to degrade the chromatin associated RNA. So this is called the exo-SC3, which is part of the exosome complex. So if you were to knock it down, then the levels of the chromatin associated RNA goes up without affecting the process of transcription. So now what we have in this system is that the levels of the chromatin associated RNA is high without the transcription being affected, and now we checked at the binding of the transcription factor. we see that after you knock down this, we see that the binding of the transcription factor goes up, and which is also shown at the genome-wide level as well. Now we know that the transcription factor, the RNA, is sort of supporting the binding of transcription factor. Now what we wanted to do is decouple the effect of two. If it is the RNA or the transcription factor or the openness, how does that work? So we took this particular mutant, that is the ER alpha mutant, which is not able to bind to RNA. So in this particular mutant, we have seen that the binding of the transcription factor to the TFF1 eRNA, which I showed before, gets perturbed, and which leads to a genome-wide loss of the transcription factor binding overall. And now what we have is a mutant that is no longer able to bind to the RNA. So in the case if you treat this particular mutant with DRB, now the opening of the particular chromatin would still be there. The polymerase would still get recruited to those regions, and which still gets stalled near those regions. But what would happen is the openness would still be there, but the RNA which is coming out of the stalled polymerase would not be able to interact to this ER alpha, because the ER alpha lacks the ability to bind to RNA. So now if the polymerase was important, we should still see an increase, but if the RNA was important, we would not see an increase. And this is what we see, that if you were to treat the RNA binding mutant with the help of DRB, we see that in the RNA binding mutant, we don't see that increase which we were seeing for wild type, suggesting that it is the short RNA that comes out of the stalled polymerase that is responsible for these effects. So saying that the short RNA keeps the transcription factor in proximity so as to regulate the binding of the transcription factor to the DNA. Now, which brings us to the question that why exactly cell is doing all of these things. So why is there an increase in the binding of transcription factor? What has cell to gain with these? So I have not shown data for another. So what we have done is we have looked at another global activation mark, which is ST cadence and acetylation, which we have seen that also follows a similar pattern, that wherever ER alpha is increasing, that is also showing an increase in ST cadence and 27 acetylation. Now what we wanted to see, how does that relate to the transcription? So what we did is we sort of allowed the transcription to happen. So this is BRUC, so which tells you how the transcription is going on, how the output of the RNA is coming out. So we did it in two different conditions. First is the mock condition, when you have not treated when you have not treated the cells with the inhibitor, and the other is after releasing the cells. So once you treat the cells with the inhibitor, so since DRB is a reversible inhibitor, you can just wash it off and the cells will begin to transcribe. Now what I wanted to see, if the regions that begin to transcribe faster were the regions that had an increase in the binding for these transcription factor, or showed an increase in ST27 acetylation. So one of the things that we see here is one example. We wanted to see if this happens genome-wide. So we bind these entire regions on the basis of how much the increase is after you release it. So 1 to 5 is an increasing order of how much the release, the output is after releasing. So what we see here is the regions that showed the highest RNA after release were the regions that showed highest gain of H2 and acetylation, or the ER alpha, the transcription factor, before. So wherever there was a higher increase in the transcription factor or H2 and acetylation, those are the regions that showed a higher release of RNA, higher formation of RNA after release. So that brings me to my model, what I'm suggesting. In the normal situation, there is an on and off rate of the transcription factor. So which the transcription factor comes to the regions and then lead to the transcription of the gene. But in situations when there is polymerase pausing, which happens normally as well, but here we are adding the drug to initiate that. And what would happen is, because of the short RNA, that would lead to the stabilization of the transcription factor. And once this pausing is released, that leads to a higher output of the transcription. So for that, I would like to thank my supervisor, Dimple, and my committee members and the lab members, which are also shown here, and NCBS TFR. Thank you. I'm happy to take questions.
Yeah, please.
Hi, Rajat here. Nice lucid talk. The mutant that does not bind the RNA, how well does it bind the RNA, the polymerase?
So it doesn't bind to the polymerase. It would not. So so in the wild type condition. So you're talking about the direct interaction of the polymerase and the transcription factor. That is what
I assume they bind.
Yeah. So yeah. No. So my bad. So in the normal wild type condition, the transcription factor does bind to the polymerase, which will recruit it. In the RRGG mutant, we have just put up the RNA binding. It would still bind to the polymerase to the same extent. That
To
is,
the same extent?
yeah, ideally. Yeah.
So, but the model that you're proposing now is that the direct interaction does not matter
Direct interaction for
between the polymerase and the transcription factor. Because you're suggesting that when it doesn't bind RNA, you don't really see the enrichment of the transcription factor, right? So then why does the transcription factor directly interact with the polymerase if it's the RNA mediated binding which is more important?
wide. So because the I think all of these interactions are happening simultaneously. So transcription factor is known to recruit polymerase to its site. So wherever there would be a motif, the transcription factor is known to bring polymerase and other to its site. That is what. And as soon as people have talked about, as soon as there is RNA coming to the mix, the binding of the transcription factor to DNA increases. That people have done it in vitro with just the reconstituted system. And another person from our lab has also done similar sort of experiments. So
I'm just hung up on the fact that why the mechanism is evolved to involve the RNA, because the transcription factor is recruiting the RNA polymerase, right? Or it's promoting the binding of the polymerase onto the DNA, right? Now, it's already there in place. So what is the advantage of having the transcription factor bind the RNA?
So in certain situations, what can happen is because of the pausing and other things. So the RNA output might not be that high. So if the RNA becomes very high on these transcribing regions, for other transcription factor, what it can do is the RNA can take away the transcription factors also. So that is another very thin, narrow range where the transcription factors can work, the RNA can work in supporting. So direct interaction and how multiple way interactions can support the transcription factor binding. How it has evolved, I'm not very sure. Why, what would be the reason? Maybe because of multiple ways to regulate the binding. That adds another layer. So why it is, but I'm not really sure.
Thank you.
So I'm trying to extract in my mind the LLPS aspect of what is otherwise a very, very fine and well delivered transcription talk. So in this model here, would I be right in saying that where you show the basal transcription, there's no condensate. Where you show the pause release, there's no condensate, and there's a condensate. No.
There is a condensate.
Go back to the model.
Okay.
Yeah. And that where the polymerase pauses, then with the help of transcription factor binding to the short RNA, the condensate is helping to keep the flock together so that it doesn't dissipate, so that once the pause is released, the transcription can resume. So is the role of liquid-liquid phase separation only in that central, polymerase pause thing?
Not really. So even in the situations when there is, like we have not induced the polymerase pausing, we see the formation of these condensates. It
So
could be.
I've heard somewhere that these transcription factories that are, hypothesized to exist, are actually liquid-liquid phase separated regions.
That is what people,
Right? Right? Not just the nucleolus, but many
other
many
transcription
regions,
factories.
transcription
Yes.
where presumably in a condensed state, transcription is continuing.
In the condensate, within the condensate, I'm not really sure. People just have done fish, which is sort of an indirect association with it. Within the condensate, what people have talked about is the smaller the size of the condensates, the higher is the transcription. That is, so if the RNA becomes too high, then that leads to the formation of a shorter
Well,
condensate.
it can't get as small as a single complex.
No, not really as a single complex. Then
So how many complexes, and are those complexes paused or transcribing?
so in these terms, people have done.
That's the key question, right?
In-
I mean, there's a fantastic talk on transcription, but to understand what is the liquid-liquid phase separation aspect of it, I'm trying to understand.
So here,
Is transcription happening inside the condensate? Or is the condensate stopping the transcription?
no. People have talked about from the so like one of these studies. Where did it go? Yeah. So here, this particular paper talks about in terms of how does the binding of the transcription factor, so if there is a formation of these condensates here, how does that relate to transcription? So people have not talked about inside if it is happening. They have plotted correlations that if it is closer to the gene, that makes it more burst. So if the condensate is closer to the gene, that makes it more burst. That is what people have talked about, not a direct correlation there. That if it happens within the condensates or not, that's not. But in vitro experiments people have done for taking condensates and then doing IVT, in vitro transcriptions, for GFPs, and those people have done that, have shown that when there is a formation of condensate, there is a higher transcription. That people have done. But in vivo, this is the best that I have seen.
Hi. Nice talk. So I'd like to, I have seen like in one of your slides, you have shown the increase in the number and size in live cell imaging, right? So is it confocal imaging? Like how did you?
So these are confocal images. These are confocal, but we have done super res as well.
Right. Because what is the size of the condensates?
So in these, so these are ensemble averages. So what I have done is I have taken the mask of all these things and combined to get the average size. So it's after thresholding. So after removing these low intensity pixels. So that is done after. But we have done super S with STED, and we still see similar things there.
And regarding the number calculation, like you have considered a volume, like how you calculate the number, or you consider single plane and
Yeah. So if it is within this single plane, and then it's just normal thresholding, and then based on, so these are intensity based thresholding, whatever is above that, I'm calling those. So, and every single frame would have a different, what I'm calling is regional discontinuity. So wherever there would be a blip, that would be called. And post that, I'm putting different size thresholds and things. If it is below a certain thing, below the diffraction limit, it would not be called. So those things I'm taking.
Because I'm just wondering, since these are very highly dynamic, they're very mobile. So when you are considering only one single plane and they are going back and forth,
so
is there any possibility of like multiple counting of the same entity?
No. So here what we have multiple counting. So this is at different frames for the same cell.
Okay.
So even if something goes up and comes back again, it would. So what we have seen after a certain time, these things become pretty stable. So if you take it for like a higher frame rate, we see that these are pretty stable. It doesn't move much. Okay. So that is what we have seen.
Okay, thank you.
So very nice talk. So in general, these ones that you're calling condensates, it's not a simple kind of a droplet that we see, right? It's a, so what is, in your sense, what is the structure? How do you physically characterize it, or have you thought about that?
So in terms of structure, meaning what are the different proteins that are associated with, along with DNA and those things?
Like a droplet, clearly.
Right?
Yeah, I mean, DNA is involved.
Yes, definitely.
So, which provides some sort of a surface.
Yeah.
Now, have you tried characterizing that? Because, of course, there are ideas that of things like pre-wetting and...
yeah, so there is a different protein called FOXA1 that acts as a nucleator for these. So, like I was talking about, that the transcription factor cannot bind on its own because it needs something to open it up. So, what we have seen is, even before the signaling begins, there are certain regions that are primed by FOXA1, so which is a nucleating factor. So FOXA1 can bind to closed chromatin and open it up. So that can assist the binding of these things. So what we have seen for that FOXA1 kind of primes it. So wherever there is FOXA1, we see the formation of these punctas are faster on those regions. So we are looking in those situations as well. And we have removed FOXA1 from this picture, and we don't see the formation of these condensates. FOXA1, we don't see the formation of these condensates. But that is also within a concentration range. If the concentration is already very high, then we still see formation. So it's within the range.
Yeah. So in general, I mean, this, like you are saying that here there is a formation of LLPS. I have seen some study, they said that when there is DNA repair, when there is the cut is happening, then also there is a formation of the LLPS, and there is some other purpose to that. My point is that all these LLPS you are forming, they are recruiting specific constituents, right? How it is even getting regulated? Is that something
Yeah, so there are a lot of a lot of studies that people are doing right now. So one of the ones that I already showed was this Med1 thingy. Yeah, this paper. So they talk about charge patterning. So they talked about that if given charge is present at a given like spacing. So only if the only for co-activators, that if it is present, the charge is patterned in such a way for if the charge patterning is similar for similar co-regulators, then it would get partitioned. So in this paper, what they have done is they have taken another different protein and made the charge patterning similar to another co-activator, and now they see that it gets partitioned. So there are people who are working on similar sort of things.
So let's thank Rajat for a wonderful talk.
Thank you.